Okay, I think we have our participants numbers for today. So we go ahead and start. So welcome everybody to today's Matter to Life lecture series. Today we have Roman Zarot, one of our fellows with us. Welcome, Roman.
You.
He's a PI here at the Max Planck Institute for Medical Research. But before we get into yeah, his personality and his way of getting to research, but also his research, I briefly want to explain you the system we're working with. So we are working with a webinar possibility, so you cannot raise your voice during this webinar, but we have this Q&A possibility at the very bottom of your screen. There's this question mark. And you can type during the talk, but also at the end of the talk, your questions within this Q&A section. And you can also upvote the questions you're interested most in. They will be then answered the first. So whenever you have questions also during the talk, please use this function to ask them, and I will read them out loud. And you can have your scientific interested answered by Roman. So thank you all for joining today. Thank you, Roman, for being here. Before I talk long lines who you are, I would ask you some questions which will introduce you to a certain point. So the first question I wanted to ask, so how did you come to the decision to join the Matter to Life faculty?
Right. I think one of, if not the most important factor in doing academic research is having excellent students, talented students. And frankly, and after just 1 year here in Germany, I've already learned that Matter to Life students are stellar. And in fact, I already have the pleasure of co-advising one Matter to Life PhD student. And I think the other thing is also that, I've only been in Germany for a year, right? And previously I was mostly acquainted with the Californian and US American higher education system. And so getting straight into this delocalized system that is Matter to Life has been an excellent way for me to get integrated and exposed in Germany and the higher education system here.
Yeah, that's true. You already got hit by all the different, also bureaucratic setups from all the different sides.
Yep.
Yeah, it has also quite some beauty in it. Okay, great. So, how does your research engage within the interdisciplinary perspective of Matter to Life?
Yeah, I think what we do, our ambition finally is to start with pretty wild new chemical biology concepts on paper and move them as close to clinical reality, as fast as possible. And my past experience has taught me that this is the most fun and the most effective when different expertises and perspectives come together from the very beginning. And so we like to integrate synthetic chemists, MDs, so clinician scientists, and people from computational backgrounds from the very get-go to advance problems and learn the most in as little time as possible. So I think, and the other extent also, I think that, the training aspect, I think training students that are not unique, let's say chemists, like I was trained myself, but instead are multilingual scientists from an early stage, equips those students to be true leaders in tomorrow's life science and biomedical science.
Yeah, that sounds really good. Exactly what we're aiming for. Okay, so if you wouldn't be a scientist, now you are a scientist, but if you wouldn't be a scientist, what would you be doing?
Training to finally win Wimbledon, probably. Yeah. Tennis player, hopefully. Yeah. Not sure I would be that close, but that would be fun to do for sure.
Somewhere on the list at least, right? Okay, great. So then I'm looking forward to your talk. I will not lose any more words, and the stage is yours.
Thank you very much. First, I want to say thank you to all of you joining today, to the Matter to Life community, and to all of you joining in. It's a delight for me to be given the opportunity to present our work here in this forum, and I'll be talking about the main focus of my nascent lab here in Heidelberg, and also some of the work that we did most at Stanford in my postdoc, which is really geared towards using small molecules to reprogramming gene expression in cancer cells, right? And I'm going to take a bit of a detour first to introduce the tool that we use to achieve this goal. And that is the fact that in almost every form of cell signaling, that is ranging from signal transduction at the cell membrane to protein degradation, protein homeostasis, to the way gene expression and chromatin structure is regulated, it is often sufficient to just bring two proteins together within a cell to activate a certain type of function. And really, we've known this, since the 90s, where seminal work of pioneers such as Jerry Crabtree and Stuart Schreiber have started revealing this really ubiquitous biophysical regulatory principle that is governing most cellular signaling processes. So bringing two proteins together in a cell is really key for us to modulate biology. And it's then also not surprising that nature has been using this forever, right? So to some extent, what my lab is trying to achieve is just mimicking, copying nature. And on this slide, this is mostly a pretty picture. You see a bunch of beautiful crystal structures of proteins, two proteins that are glued together by a small molecule in orange. And these molecules mediate their pleiotropic biological effects exactly through this mechanism that I just mentioned. And that is through bringing two proteins together in a cell that usually do not interact, right? And that's what we try to do. And we try to do it, as I mentioned on the first slide, in the domain of modulating gene expression. Now, gene expression, of course, first step in the central dogma of biology. And we want to do this in a cancer context. The reason being that, cancers have different mutations, different cancer drivers, different enzymes that drive this aberrant proliferation and growth that is characterized as the hallmark of cancer. But they have one commonality, is that all these different mechanisms converge on changes in gene expression. And really, this deregulated gene expression mechanisms are at the very core of cancer cells. Meaning that if we have approach a drug, let's say, that could directly target this aberrant and deregulated transcription in cancer, if we're really going directly for the jugular of cancer, right? It's the most direct we can go for a cancer cell when trying to eliminate cancer as a disease. Now, the problem is that at least when you're a small molecule chemist, such as is my background, we're pretty limited in how we can do that, right? So in the past 50, 60 years, how medicinal chemists have addressed the problem of drugging oncogens, so proteins that drive the growth of cancer, they have fallen under the paradigm of what I refer to as occupancy-driven pharmacology, and some of the molecules that adhere to this principle are shown on this graph. In general, they bind the protein, often enzymes, and try to inhibit it. So they just try to shut off the activity that is driving the cancer, which of course is a very intuitive way of thinking about drugs in that context. The problem is that most proteins in cells actually cannot be addressed like this for reasons I don't necessarily need to go into. Well, actually, they're just not enzymes. And then in the 10 years, or maybe 20 years, last 10 to 20 years, there's another pharmacology that kind of took center stage. These are molecules that don't just bind to inhibit an enzymatic effect, but they fully shred, they destroy these proteins. But you can imagine that while mechanistically distinct, these molecule modalities finally do the same thing, right? They phenocopy and knockout. They try to silence overactive protein function trying to slow down tumor growth, but they're pretty bad at killing cancer cells, as it turns out, actually. In addition, a lot of these drivers of cancer growth are actually also important in healthy cells. And so if you administer systemically a drug that inhibits their function or gets rid of them fully in a clinic, you imagine you will have toxicity effect in healthy cells, and so horrible toxic effects in patients as well. So we want to take a fundamentally different approach to inhibition or degradation and silencing. We want to do not loss of function, but gain of function. We want to activate and not inhibit. And the paradigm is very nicely illustrated here, right? So those cancer drivers that we had before and that people have been trying to silence or inhibit, we want to instead take the activity off and connect them to cell death pathways that are already genetically encoded and evolutionarily perfected in every cell, but have been silenced in those. Reactivate those pathways and potently and hopefully specifically kill cancer cells. And of course, you want to do this at a transcriptional level. And you can already see just with this graph, right? So we want to do this with a molecule that has two sides, a so-called bifunctional small molecule, an inducer of proximity, of protein proximity. In other words, we tried to use this biophysical regulatory principle that I introduced in the beginning, just bringing two proteins together to induce signaling that doesn't occur without those proteins talking to each other, and actually not just induce proximity of two proteins, but actually make two entire pathways, cancer driving and programmed cell death, come together and form a new interaction. Now, cell death is powerful in cancer. And in fact, cell death is important for all of our cells in higher development. All of us right now lose about 1 million cells per second in our bodies. And obviously, we're pretty, most of us hopefully are doing well and don't feel any of these effects. There's no resistance, there's no inflammation, right? And these are really highly perfected mechanisms that get rid every cell that is just a bit weird. Cells that if they persist, will develop in something worse. So our body is really good at getting rid of these cells through programmed cell death pathways. The most programmatic one is called apoptosis. There's others as well. But of course, cancer cells are, difficult ones. They have found ways to overcome these mechanisms and to silence them. And really, the goal that we're trying to pursue here is to reactivate those very perfected killers, in this case, to the detriment of the cancer cell. Now, how do we realize this? This is kind of the picture, the thinking picture that we have in mind. So we try to develop molecules, again, these bifunctional molecules shown here, that bind on one side to a transcription factor that binds to DNA with sequence specificity and ideally represses genes that, when activated, kill cancer cells. Oops. And on the other hand, we recruit a transcriptional activator that usually drives the proliferation of cancer cells. And the molecules that result from this, I'll be referring to them a lot like that, are called TCIPs, or transcriptional chemical inducers of proximity. And, in many ways, this yellow transcription factor here is just a sequence-specific DNA binder that sits at the right genomic loci for the gene sets that we try to activate. And because we know that transcription is inherently regulated by induced proximity, we can already kind of guess that if you just bring the right activator to the right gene, it will induce their expression. And importantly, we do not want to do any modification, genomic modifications to the cells. We want to do this with just acute, reversible, small molecule pharmacology, meaning that if we find something, it's directly translatable, possibly into mice, possibly into humans. Now, it's important to note what TFA, so this yellow protein, is that we used in previous work. And that was, we focused on a transcription factor called BCL6, shown here. And BCL6 is a nice one because it is one of these master regulators of cell death. It binds DNA sequence specifically at promoters of so-called pro-apoptotic genes, which, when expressed, kill very proficiently, but then recruits a host of co-repressors here in blue that, when recruited, then have a silencing effect on those genes. And BCL6 has a very important role in our autoimmunity, actually, so in immune response. But certain cancers, specifically B-cell lymphoma, diffuse large B-cell lymphoma, hugely overexpress this factor and ensure that they're essentially immortal and very resistant to other oncology treatments. You cannot knock out BCL6 from these cells. If you do, cells will just die. And you can imagine that for that reason, BCL6 has been considered a very attractive target for pharmaceutical discovery for a decade or so. And actually, and this is one of my favorite molecules ever, developed here in Germany, actually, by Boehringer Ingelheim, BI 3812 and others. They bind BCL6 at a structured domain very potently through a mechanism that, the standard way of how we have been thinking oncology drugs, which is just binding to silence, right? And they bind very potently in the nanomolar range. But turns out this just doesn't have the effect that is desired. Just binding, inhibiting this co-repressor complex does not have a strong effect on cancer cells. They don't die, except for at very high concentrations, never made it into humans. For us, of course, perfect scenario, because we could now develop bifunctional molecules that don't just bind to BCL6, but in addition, recruit an effector, an activator, a cancer-driving activator to those loci pre-bound by BCL6, activate gene expression exactly there, and because those loci are pro-apoptotic, kill lymphoma cells very potently. And we have done this, across a few examples. My postdoc lab previously had shown this works for elongation factors, p84. And the topic I'm going to talk to you about right now is my own work, which focused on transcriptional kinases, CDK9. And in the meantime, we have already worked on additional activators that leverage different layers of chromatin regulation. So it's really emerging as a general pharmacology. And since this is a really new and I guess foreign concept, it sure was to me when I started working on this. I want to quickly highlight what we think these molecules are doing, right? So here's another picture of BCL6 sitting where it likes to sit on DNA, right, at those genes that kill when expressed, and it binds the co-repressor complex. In this context, in this state, transcription of these genes is shut off. The cells are doing peachy fine. This is like you're sitting in the car and you have the brake fully pushed down, so it's still. Now we have the BCL6 inhibitor from Boehringer Ingelheim that disrupts this co-repressor complex. This is apparently not sufficient to induce gene expression. This is like taking the foot off the brake, but that is of course not sufficient to accelerate the car, as those car drivers among you know. That is exactly what TSIPs do. So TSIPs now, in addition, harness the transcriptional activity of an activator, and that is now putting the foot on the gas, on the acceleration, and that then drives gene expression and cell death in these B-cell lymphoma cancer cells. Okay, and now let's go into the actual exam. So my work focused on this transcriptional kinase CDK9. Kinase, of course, are phosphorylating enzymes. CDK9 is a ubiquitous activator in every cell, and it is known to having to phosphorylate the so-called C-terminal domain of polymerase II, which is the main transcribing enzyme, right? And this phosphorylation basically activates polymerase towards moving into the gene body and activating transcription. CDK9 is involved in sustaining a pro-proliferative gene program in many cancers, and many of these molecules that I've shown you before, actually, and some are repeated here, have been developed to inhibit this function to shut down oncogenic gene activation. The problem is, because CDK9 is essential in healthy cells, this has been met with toxicity in the clinic, and these molecules have not been advanced further. We, of course, try to harness this activity and turn it against cancer by recruiting it to BCL6. And how did we go about this? Well, first it started with chemistry. We take a chemistry-first approach. Synthetic chemistry usually is first for us. And so we had to make bifunctional molecules. And a general scheme is shown here, right? So we took all these CDK9 binders that are known, almost all that are known to man, and linked them up to BCL6 binders that on their own don't do anything to cancer cells. And the special sauce, if you will, is in this linking motif that connects the two binding partners. Despite great advancements in silico predictions of of protein complexes and dynamics, we cannot, and to my knowledge, nobody can at this point predict which of these linkers, what composition, what length, what rigidity will do best at forming a good ternary complex in cells, right? And so we have to make libraries, and that's exactly what we did. And then we generally rely on a suite of robust cellular and biochemical assays that are to be that we use to address questions such as, do any of these compounds actually activate BCL6 dependent gene expression? Do they do this in a manner that is dependent on bringing the two proteins together in a cell? And hopefully, does that translate into very potent killing and specific killing of cancer cells? And we know at that time we made about 100 compounds, which I hope we don't have to do again. That was a lot. And I'm skipping a lot of work here, but we converged on a lead compound here called CDK-TSIP1 that connects this Boehringer Ingelheim BCL6 binder with a CDK9 inhibitor. In this case, it was a C7 spacer in between. And we always make these chemically matched and approximately same molecular weight negative control compounds that, through small modifications shown in red, lose the capacity to bind one of the binding partners. So they serve as negative control compounds that get into the cell approximately the same as the lead compound. And this is probably still one of the more exciting results of the career in my last 5 years or so. We use this reporter cell line where we have a plasmid encoding for green fluorescent proteins under the promoter of BCL6. We lentivirally transduce this into a lymphoma cell line that has a lot of BCL6. And anything that now binds to BCL6 and de-represses genes downstream of it will lead to an increase of green fluorescence. And that is exactly what we saw with our compound in red, whereas the two negative controls did not have this effect at all. So it seemed like, wow, we've been able to turn a CDK9 inhibitor, the SNS032 compound, into a locus-specific activator of transcription. That was completely unheard of. Still very excited about this fact. And we tested in a biochemical assay that this compound really is able, this is a TF-RED assay with expressed and purified protein, that these compounds can bring CDK9 and BCL6 into close proximity, whereas negative controls by design cannot. And you will see this characteristic bell-shaped curve, right? That is a hallmark of bifunctional inducers of proximity, because at high concentrations, you get saturation of binary binding, which competes with ternary complex formation. So directly, we stipulated and we had a strong feeling that this whole effect is driven by a cell-active and transcriptionally competent ternary complex. And indeed, it did also, that's probably most excitingly, translate into very potent cell killing of a BCL6 overexpressing and dependent lymphoma cell line, SUDHL5. And this is a 3-day assay. That is our workhorse assay, so to say. We found a single-digit nanomolar EC50, very potent killing. And importantly, look, you should consider the window, the difference in potency to the black line, which is the co-treatment of the parental inhibitors of what constitutes CDK-TSIP1. And that is despite those small parental inhibitors getting into the cell much more potently than the large and floppy and not very permeable bifunctional molecule, right? And that is where we were starting to think, okay, probably we found something special, because obviously very little of this compound gets into the cell and still drives a much more potent phenotype. And that's what we think is this gain of function effect that we're drawing upon. Now, when in cancer, right, potency is only one side of the medal, though. Specificity is another, right? We don't want those toxic side effects that many patients have in a clinic. And that is very hard to assess in an academic setting. And so what we did do in this setting is that we used lymphocytes from patients from a hospital that are healthy cells, right? And just compared how toxic are other CDK9 targeting drugs in this cell model, and found that a very potent CDK9 inhibitor, for example, is still EC50s in a nanomolar range. Degrader that gets rid of CDK9, also very toxic. SNSO32 toxic still, and our compound loses about 3 orders of magnitude in cytotoxicity when going from a lymphoma line to a healthy line. So we got a bit of a feeling that, okay, we might have discovered something that is not just toxic, but maybe also lineage specific. And indeed, that's what we found when we went into a bit more detail here. So this is work that we did in collaboration with the so-called PRISM consortium at the Broad Institute in Boston, where they have the capacity to screen drug sensitivities in almost 900 cancer cell lines at once. And I don't want to go into too much detail, but basically each dot is a different cancer cell. They're ordered cancer cell line. They're ordered by lineages. And the bigger the pink bar, the more sensitive our cells to our compound. And I think it's relatively clear that it's really these two lineages here, which are those lymphoma, non-Hodgkin lymphoma lines that are epigenetically deregulated in BCL6 that are sensitive to our compound. And so indeed we found something that is not just very potent, but also very specific, right? And that's kind of where we want to go to, right? In all of our projects, we hope to have potent and specific effects. And once we do, we like to go really deep in mechanism. And that's also where the interdisciplinary aspects come in even more, because then we go into involve epigenomic and transcriptomic effects. We look at, is this ternary complex really formed on chromatin? So what I'm showing you here is so-called ChIP-seq data. So that's chromatin immunoprecipitation and sequencing. Basically, we shred DNA, pull with an antibody, and then sequence the pieces of DNA bound to those proteins to find what proteins and how much of that protein is bound at a given sequence before and after drug treatment. And we found when we did this ChIP-seq with a CDK9 antibody that our drug leads to specific and dose-dependent recruitment of CDK9 to loci that are pre-bound by BCL6. And when we then do another set of experiments that use an antibody for polymerase 2, we found that those sites that have more CDK9 now, shortly after, also have much more polymerase 2, meaning activated transcription. So we're able to show that our compound really first recruits CDK9 to BCL6, and that this then activates polymerase. And you can imagine what follows then. If we activate the transcription of genes that are downstream of BCL6, we of course kill cancer cells very potently. Now, of course, the question was, we go back to chemistry, because chemistry remains essential and center stage. We wanted to ask, that's beautiful in cell lines, cancer cell lines, but cancer cell lines are never reflective of patient samples and of the in vivo setting. And so we would have really liked to go in vivo. The problem is that our initial compound, CDK-TCIP1, had no stability in mouse plasma or in human plasma. It just, disintegrated. And that's relatively common for these large and floppy bifunctional molecules. And so we did, more chemistry added on top to those 100 compounds we already did. But in this case, worked what is very common in the targeted protein degradation field, and did a focused medicinal chemistry campaign focusing on this linker motif. Generally, the goal is to rigidify the linker, to decrease metabolic liabilities, to enhance cellular permeability and also solubility through the incorporation of basic amines. And as you can see, we relatively rapidly and robustly identified compounds that are even more potent, in some cases sub-nanomolar EC50s, again, 72-hour assays, that go into the cells and activate BCL6 transcription more potently, and now have half-life times in mice that are at least in the hour range. This is still not very stable, by the way, but was okay to go into mouse experiments given how potent they are. And that's exactly what we did. We did this in collaboration with Michael Green's lab at MD Anderson in Texas in the US. And we actually chose to do something more general than a tumor model right away, even though we have done that in the meantime as well. And I told you in the beginning that BCL6 is a master regulator of cell death and important in our immunity. And actually, its activity in suppressing cell death is really important in those B cells, the germinal center B cells, that produce antibodies after infection, right? If BCL6 were not there, the cells that try to make antibodies for us would just die in the process. And so we said, okay, if that's true and our molecule kills off cells that have high BCL6, then we should see that in a mouse immunization model. So in short, we injected a mouse or mice with sheep blood as an infection. And then after a certain amount of time with and without a drug, looked at the FACS, so flow cytometry, whether we killed off these terminal center B cells. And that's exactly what we found here to the right. We found a dose-dependent ablation of the terminal center B cell population. You can also see in dark green the most potent effect we found in a twice daily dosing regime. This is, of course, because the compound still is not that potent, stable, sorry, it was degraded over time. But here comes the exciting thing. This effect was completely specific, and we saw no effect on other B cell populations, such as naive B cells and memory B cells. And for all those of you that are clinically interested, this is exciting because all other B cell targeting therapies, including cell therapies, they get rid of all B cell populations. And actually, if you look at CAR T cell therapies, after relapse, the most common reason for patients to die is infection because you got rid of their entire immune system. Possibly here, if we can retain large parts of the B cell population, maybe the immune system could come back faster. Much in the future, of course, but God is very excited about the approach. And I'm almost, trying to wrap up. I'm going to tell you a bit what we're doing right now. So, this work that I just told you about, right, focused on the recruitment of transcriptional kinases to BCL6. I told you that currently we have work under review where we try to recruit acetyltransferases. They transfer acetyl residues to histones. They regulate openness of chromatin, so it's an entirely different activation mechanism than CDK9 or PLE4. And surprisingly, we see very distinct effects in what gene sets are activated. And that suggests, of course, that possibly we can harness different layers of gene expression control to create very specific molecules, different and maybe even access and have effects in different cancers. And we think that is because these molecules, and I'm getting back to the very beginning, are mirroring the natural way gene expression is regulated in cells. We've also established these molecules as proof of concept studies for transcription factors where, unlike BCL6, Boehringer Ingelheim hasn't already gone and made beautiful molecules for. So here we've been looking at an oncogenic fusion, EWS-FLI, which is a strong activator in a really horrible bone cancer in kids, and have shown that this approach may be able to reactivate tumor suppressor P53, at least one mutant thereof. So this is already going beyond BCL6, of course. And as well as a teaser now, what is going on right now in my lab, maybe at some point with some of you in the Matter to Life cohort is obviously, for a given repressor, we have used CDK9, we've used PLE4, we've used P300. There's a whole host of additional epigenetic activators, cancer-specific, non-specific, that we are exploring for new effects, for new specificities. And the big goal here is to fully leverage the potential specificity potential of these molecules, which is, of course, logic and gating. So if both Blum proteins are fully cancer-specific, only expressed in cancer cells, we can get the most specific effect in cancer cells. But as already kind of hinted at, many transcription factors do not have small molecule ligands yet. So many of the transcription factors you'd like to rewire with induced proximity don't have that. And so we're going that way. That's a hard and long way. We're trying to find new drugs, new small molecules and peptidic binders to access new transcription factors and new cancers for this pharmacology. And actually, this project is led by a Matter to Life student who's making absolutely awesome progress. Her name is Rafaela Küppers. She will be on the next slide as well. Then you can imagine that instead of activating, we could also think about recruitment of repressors to activators. That's no longer gain of function, but has a lot of intellectually interesting questions that are buried in this scenario, thinking about several nuclear hormone receptor-driven cancers. Think about breast cancer, for example. And finally, we're going even beyond transcription. What I really like about transcription is that DNA serves as a template. It reduces the problem of 3D, bringing into 3D proximity, bringing two proteins together in 3D to 2D, because DNA forms a template. And so another DNA templated process that I like is DNA damage repair, where we start to recruit things to repair enzymes to hopefully induce cell death, specific cell death in different cancers with high specificity. And so what I hope that you take away from the past 40 minutes is that why chemically induced proximity, I'm sure you have heard a lot about it, but I'm sure you have mostly heard about it in the context of targeted protein degradation. But I think that's really just the tip of the iceberg, and we're bound to explore and find much more that is right now still hidden under the surface. That's not just gene transcription at all, of course, but that's the domain where we're chiefly interested in. And in the years to come here in Heidelberg, I hope that we can push away from just cell death, induction, and lymphoma to new cancers, and to the gain of function, activation of other phenotypes as well, being to dosage restoration in so-called haploinsufficient diseases, inducing differentiation or resensitizing tumor cells from an immune cold to an immune sensitized environment again. With that, I want to thank especially my lab. Shown here is the first lab picture we had at the past Christmas, and all the team members that are doing the hard work. It's really exciting to work with them. I want to thank Matter to Life for accepting me into their program. It's really a delight to work with them and the students that I've already had the pleasure of hosting, and looking forward to discuss, maybe meet some of you and answer some of the questions. Thank you.
Thank you, Ruben, for your talk. So everybody who is interested can put some questions now in the Q&A section before I give you a bit of time typing. And so I'm a trained chemist by myself, so all this biology part is very off my comfort zone. So sorry if I ask a very stupid question, but this BCL6, does it have also natural activators which might concur with your binding of your molecule? And or is it not so prominent, especially in the cell line you have been using for testing your compounds?
Just to make sure I understand, you're asking whether BCL6 has endogenous activators.
Yes.
Yeah, so P300 actually is a known interactor of BCL6, and I think that's part of the reason why these molecules that recruit BCL6 work so well, because they basically enforce a weak pre-existing interaction. But usually the repressors have to be displaced before that can happen.
Okay, so okay, so they're endogenous also expressed in the cell, in the normal cell at least.
Yeah.
Okay.
So far we have actually, yes, they do, but they're just not interacting or only weakly interacting. Yeah, we're not adding anything to the cells. Everything is there, just rewiring. We're mechanics forging new cables.
But your molecules you're still adding.
Yeah.
Right? Yeah. So these are, so I mean, they look really complex molecules. So how is the synthesis? How many steps do you have? Is it really a pain in the ass to make this library? Library is always pain in the ass, but like it looks like complex synthesis for me when I look at the molecule. How is that?
I think they're actually quite fine. I think it's in a volume. If we have to make 100, then it's in the sheer volume, and we're working on technologies to make this more high throughput. The synthesis depends a bit on your training, right? For a trained chemist, I think the complexity is manageable. It depends a bit on whether the binding motifs, whether they're already commercially available, then just linking attachment and connecting is easy. The purification can be very challenging. These compounds are very insoluble, so that is challenging. But I would rate that doable, and in fact, is an excellent training ground for new people in chemistry, and I've had several students here in Heidelberg who picked up that very effectively.
Okay, great. So you have a stone at the bottom of your flask. How to solve it again. Okay, great. So now we also have two questions from the audience. So I would just go ahead and read the first one. TF is hard to bind with. How do you identify a ligand?
Yeah, so this is this paradigm of transcription factors being undruggable, right? So I hate the term undruggable anyway, but I always refer to it as not yet drugged. I think first, they're not enzymes. And so when you just want to do enzyme inhibition assays, of course, you will not find ligands for transcription factors. But then there's transcription factors and transcription factors, BCL6 and where it belongs to. They bind DNA, they're transcription factors, but they also have nicely structured domains that have pockets. And they're difficult targets, but I think they have mostly been not liganded because people haven't looked and not because it's impossible. That is different to like fully unstructured transcription factors, such as MYC, for example. Now, how to develop them? I think biophysics first approaches, really looking for just functionally agnostic binding is the way to go. So we can screen libraries by surface plasmon resonance, and that has worked. We can, of course, use DNA encoded libraries, so just kind of grow and scale. That certainly is promising. And finally, and that's what we're using in the lab with Rafaela, computational peptide design, right? That has certainly advanced discovery of new binders tremendously. It is much faster than small molecule ligand discovery.
Okay, thank you. So we have one person asking, Gabriele is asking a lot of questions. So I will start with the first, then continue with a chain of questions. So the first one is there an off-target linking to your linker?
I'm going to assume this is in the cell. We thought about this a lot. So especially for the BCL6 ligand, we did not know whether this is a specific binder and whether it binds other proteins. And then we did mass spec-based proteomics and actually found that it is very specific and only very selectively forms this ternary complex between BCL6 and the activator that we're trying to recruit. So I think off targets, probably quite a few, not that many. Of course, at high concentrations, the molecules will start behaving as a BD4 or CDK9 inhibitor, and that if you dose it too high, then you will see on-target toxicity because CDK9 remains an essential protein in cells, in healthy cells as well.
Okay, so then the second question would be, how do you deliver the linker in the cell, I guess?
Deliver?
Yeah. So how do you bring a linker
so,
in the cell? Yeah.
So the synthesis is done outside, right, in the hood. We don't assemble the molecule in cells. Turns out, these molecules get into cells. They're very big and they do not conform to what was used to be called the rule of five rule for the design of small molecule drugs. And I think we can quantify actually how well or how poorly they get into cells, and they get into the cells orders of magnitude less effectively than regular small molecules, but because they're gain of function, because they're activating an effect rather than having to saturate an enzyme, that is still potent, that is still enough to drive a much more stronger cell killing effect. So I think that's the power of accessing new pharmacological mechanisms with this technology that we don't even need and maybe don't even want too much of the compound in the cell.
To prevent worse. Okay, and the third question would be, can the linker be harmful itself to a cell or a normal cell, if you want to be?
So,
the linker itself.
Yeah.
Yeah, can it be itself harmful to the cell, so without its activating function, I guess is meant here.
The linker is certainly not innocent. In an ideal scenario, the linker is participating in a cooperative interaction that really glues the two proteins together. So then we, with biophysical methods, we like to quantify so-called cooperativity, meaning that forming of a ternary complex is actually favored over just the binary interactions, and that happens then if new actual protein-protein interactions are formed. And in all these structures, usually the linker forms interactions with the proteins as well. The linker itself will, at some point, is often a metabolic liability. It can get oxidized. And it can be, in certain cases, that those oxidized forms certainly have toxicity on their own. And that's exactly why I don't want to make strong claims about lack of toxicity in our preliminary academic studies, right? That toxicity studies are very expensive, very lengthy, and usually reserved for industrial setting. So they can be. We don't have at this point indications that they are.
Yeah, and will take like 8 years till you get there, right? Yeah. Okay, great. So there's another question. Can CIPs or CIPs be targeted to specific cellular compartments?
Yes. First, a lot depends on where the proteins are that you're trying to rewire and bring together. There's Professor Bannock at Stanford University his program centers around that, using CIPs to shuttle proteins between compartments. So this is an emerging topic. I would say that most of these molecules will have an effect on localization of proteins. Think about BCL6 as really strongly associated to chromatin and bound rather strongly. Whatever we link up to that will probably, if it has a persistence also in cytosol, lead to a recruitment preferentially into the nucleus. So we also need to start looking a bit more into the microscope, what is actually happening. And luckily here at the Institute, we have pretty strong expertise in just that. So I hope to do that very soon.
Okay, great. We have another follow-up question to the first question. So the first question about the binding of TF and how you would identify ligands. And now it's a follow-up question. How would you actually screen for binding ligands?
Yeah, so with these, so of course it depends on the protein you're interested in. Let's say if it's BCL6, it has beautiful domain structure. We're just interested in the protein-protein interface domain, the so-called PTB domain. Turns out that one it's very nice and easily expressed in E. coli. We can purify it well. We can clone it with different tags that allows for immobilization on chips, or for example, chips for surface plasmon resonance, and then you just have libraries of small molecules flow over that chip and measure resonance and see whether there's an interaction. Or you incubate with a large number of barcoded small molecules, such as in DNA encoded libraries, pull down and see what's stuck to the surface. Of course, because we're interested in CIPs, we could directly look for ternary complex formation, but then you have to choose what effector you wanna screen for. But I think SPR, BLI, and DEL are probably some of the most promising, and we're doing those, so those work. Say that.
Okay, thank you. So for now, one last question. So Christoph is asking, superb talk, thank you. For TCIP drugs, it seems that there's a balance between potency and permeability, stability that needs to be found. So 2 questions. It's actually 2 questions. Could you comment on the possibility of alternative drug delivery systems that could help regarding these issues?
Yes. The balance certainly with stability is there. So I showed you this hook effect, this bell-shaped curve, meaning if we dose too highly, we'll shoot over, right? And that will be detrimental to the therapeutic effect, and we might just have inhibition and toxicity. We dose too low, then we don't have an effect either. So that complicates things. Metabolic stability, actually, in the meantime, we've already done this. We can really optimize these to be as stable as clinical drugs. So metabolic stability is not a problem, I would say. That's very addressable. Actually, clearance becomes an issue because these are much bigger molecules. They are actually secreted and degraded in different pathways and organs than other orally bioavailable drugs. And delivery methods. Okay, so whoever's asking this question is maybe interested in ADCs and antibody drug conjugates. which is, where you fuse a small molecule to an antibody that recognizes a cancer-specific surface marker and then kind of leads to specific delivery to cancer cells that can increase specificity, but also, of course, increases challenge in the design and the stability of the antibody and cost. I think, and the industry is pursuing this a lot for small molecules in general, but also for protein degraders. So that's certainly possible. I think personally, the beauty of TSIPs and SIPs is that they can be already so very specific without that, and are so simple and possibly cheap to produce and thus accessible to a bigger patient population. That's an advantage we will lose as soon as we go to ADCs. But it is an interesting venue to go after. And then, of course, all these lipid nanoparticles and vesicles that we could use to bring more of a TSIP into a cell. I think that would work. But for the TSIPs we currently have, I think we might actually run into toxicity concerns in healthy cells, because in those we just, the molecule we go into more healthy cells and cancer cells, but only in cancer cells is there BCL6. But if too much of the molecule gets in a healthy cell, one of the ends inhibits a essential protein, and we're back into on-target toxicity land.
Yeah, great. Okay. And the second question here, Christoph is asking, could TSIPs become combined with antibody drug conjugates to enhance tumor specificity? Whatever the word,
Yeah, so that's basically what I said before. Yes,
Yeah.
they can. Perhaps they will. I will not do that preclinical or clinical development. I think from a chemical biology perspective, it will probably be more interesting to start recruiting really cancer-specific activators rather than staying with the system that we have and make specificity, but also complexity and cost higher by fusing to an antibody. But I think it will work. So if somebody wants to do this, I think it will work. That I can say.
Okay. Yeah, I noticed in the moment I read out the question, that's actually already was answered. Okay, thank you. So that has been the questions from the audience. You have now, 20 more seconds if you still have a question. In this time, I will announce next week's lecture talk. Next week, we will have also one of our newer fellows, Laura Heinen, with us. Next month, so in April. So stay tuned and check out her talk. And as there are no more open questions coming now from the audience, I thank you all for listening. Thank you, Roman, again for this really nice talk. And see you all next month then to our next participant, or speaker, Laura Heinen of the MTL lecture series. And have a nice afternoon. Bye.